Okay, good afternoon, everyone, and welcome to the second edition of ALACI Seminar Series. My name is Irving Martinez, and it's a great pleasure to welcome you on behalf of the ALACI junior community. The ALACI Seminar Series was created as a space to promote scientific exchange, viability, and collaboration across the immunology community in Latin America, the Caribbean, and beyond. So we are very pleased to continue a series today with two researchers working on different and complementary aspects of immune regulation. Before we begin, we invite everyone to join us through the YouTube channel to submit questions in the live chat throughout the presentation. Our organizing team will collect them, and we will address as many as possible during a joint Q&A session at the end of both talks. We will also share an attendance and feedback form in the chat, and we will great appreciate your response. Each speaker will have approximately 20 to 25 minutes for the presentation. And with that, it's my pleasure to introduce the first speaker. Our first speaker is Dr. Aracely Perez-Lopez. Dr. Aracely Perez-Lopez is an associate professor at the Biomedical Research Unit of the Faculty of Higher Studies Iztacala, from the Universidad Nacional Autónoma de México. She was trained as a pharmaceutical biological chemist at UNAM and later completed both her master and PhD in molecular biomedicine at Investad. She then carried out postdoctoral research training in Dr. Manuela Rafatello's laboratory at the University of California, Irvine, and the University of California, San Diego. Her research focuses on host microorganisms interaction and the mechanisms that drive acute and chronic inflammation. Her work has been recognized with distinctions, including the UC Mex Osconacid Monchar Award, the American Association of Immunologists Ron Young Investigator Award, the Mucosal Immunology Study Team Scholar Award, and the 2025 Immunology Award from the Mexican Society of Immunology in Basic Immunology. Today, she will present us the talk entitled CCR3 Revisited, New Insights into Mucosal Inflammation during Respiratory Infections. Dr. Perez-Lopez, thank you very much for joining us, and all yours. You can go ahead.
Hi everyone. Thank you very much for the introduction, Irving, and thank you very much to the organizers for this kind invitation. It's a great pleasure to me being here today. So I'm just gonna start talking about what we know, like classically, about CCR3. CCR3 is a chemokine receptor that belongs to the CCR family. It's coupled to G protein signaling pathway, and it has several ligands, including CCL11, 24, 26, 28. So we know CCR3 basically because it's a very well-characterized receptor that it's involved in the recruitment and the activation of eosinophils to the airway, for example, when we have any kind of allergic response. So in addition to recruitment to the airway, CCR3 is also activating the eosinophil. So when CCR3 binds the ligands, then there is a trigger in several signaling cascades that, of course, involves the chemiotaxis, but also other pathways that are involved in inflammation in the tissue. For instance, eosinophils can induce the respiratory burst and also produce some proteins that are involved in tissue inflammation. So for instance, here we have this study where the CCR3 role is highlighted in these eosinophil functions because there is an allergen model in the airway. So if CCR3 is present, then we have some damage on the cilia. We have also edema and, of course, eosinophil infiltration. However, in absence of CCR3, then we have a more preserved structure of the epithelium. And this is the role that classically has been studied about the CCR3 function. But then we wonder whether CCR3 could function beyond its canonical role in eosinophils. And I'm going to tell you why this story started some years ago. And we weren't actually focused on CCR3. We were focused on CCL28, which is one of the ligands for CCR3. And at that time, we were studying what was the role of CCL28 in the defense against the bacterial infections in the gut and in the lung. So I'm going to focus on the lung because all the work I'm going to present has been done in the lung. So basically what we found was that when we infected mice, wild-type mice with Acinetobacter baumannii, this is a very acute infection in the lung, we have an increase in the recruitment of cells to the lung, which indicates inflammation. However, in the absence of CCL28, we found this significant reduction on the presence of the cells on the body. So we asked, what were those cells? Actually, Acinetobacter baumannii infection is characterized by a massive neutrophil recruitment. So here we analyze the lungs of those mice. And as you can see here, the wild-type mice, uninfected mice, basically, they don't have neutrophils. We identify neutrophils using an anti-lytic G antibody. So we observe a massive recruitment of neutrophils in wild-type mice. However, there is a significant reduction on neutrophil recruitment in the lung of CCL28 knockout mice. So that was a surprise because then we saw reduced neutrophil recruitment on these lungs during the infection. So our next question was whether neutrophils express CCR3. And I'm gonna just mention briefly how neutrophils are canonically recruited to the lung. We know that neutrophils are produced in the bone marrow, then they go to circulation, and in circulation, they upregulate the chemokine receptors CXR1 and CXR2, and those are the responsible mostly for the arriving to the inflamed tissue, the lung, the skin, or the gut. But what we have found recently, or there is growing evidence about that once the neutrophils get to the site of the infection, they actually downregulate CXCR1 and CXCR2, and then upregulate other chemokine receptors, for instance, chemokine receptors from the CCR family. And we ask whether CCR3 can be upregulated in the neutrophils when the neutrophils were recruited to the mucosa. So to answer this question, we took those neutrophils from the valve from infected mice with Acinetobacter baumannii, and we evaluate the CCR3 expression on those neutrophils using flow cytometry. We got it from the bronchoalveolar lavage, from the blood, and from the bone marrow. And as you can see here in this histogram, we see low levels of CCR3 in the neutrophils from the bone marrow. However, most of the neutrophils are expressing CCR3 when are located in the lung. So we wonder whether this wasn't a specific phenomena occurring during Acinetobacter baumannii, or it was a more general phenomena that occurs during lung inflammation. So in the lab, then we tested other two models of inflammation in the lung. We tested the influenza A virus infection and also the LPS-induced ARDS model. And again, in both cases, we saw very low levels of CCR3 in the neutrophils that are coming from the bone marrow. However, when those neutrophils are located in the lung after infection, then a vast majority of these cells are expressing the CCR3 receptor. So here the question is we know that for the neutrophils to arrive to the lung, CXCR2 and CXCR1 are required. So why neutrophils need to upregulate other type of chemokine receptors once they arrive to the lung. So we performed some studies in vitro to try to evaluate whether CCR3 was used by the neutrophils for recruitment or actually to induce some effector functions on the neutrophils. So we did some bacterial survival assay. We purified neutrophils from the bone marrow, put in contact with bacteria, and added either CCL28 or CCL11, which are ligands for CCR3. And as you can see, about 90% of the bacteria can survive with the neutrophils alone. However, when we stimulate those neutrophils with CCL28 or CCL11, you can see how the percentage of the bacteria that survive is reduced significantly. So then we evaluate whether this was due to the CCR3 receptor. So to evaluate that, we, in addition to add the ligands, we added a CCR3 antagonist, which is this small molecule called SV328437, that from now on, I'm just gonna call SV, or the CCR3 inhibitor. So here we have the reduced survival of the bacteria when it's treated with CCL28. However, in presence of the inhibitor, you can see that we reverted this effect, both in the case of the CCL28 and the CCL11. So it seems that when we stimulate CCR3 endoneutrophils, then the neutrophils can boost the effector functions. In the case of the influenza infection, again, we put in contact neutrophils with influenza virus. So when these cells in contact with the virus, you can see here some extensions. That means that neutrophils are forming nets. But those nets are more prominent when we added CCL28. But again, when we inhibited this receptor, you can see pretty much how the nucleus of the neutrophils are condensed. So basically, we inhibit the ability of the neutrophils to produce those nets by inhibiting CCR3. So those were like some experiments that indicate us that CCR3 are probably for recruitment, but also to activate the neutrophils. So we had those studies in vitro, and then we wondered, like, how does CCR3 regulate the immune response against influenza during an in vivo infection? So to test this, we use our infection model with influenza virus. We inoculated mice intranasally with 1 to 10 to the 6 plaque-forming units. And then we gave several doses intraperitoneally with the antagonist and performed the analysis at different time points indicated here in the figure. So one thing that we do to monitor how the infection is progress in the mice is just weighing the mice and see how much they change in their weight. So as you can see here in the red line, mice that are infected with influenza, they can lose about 20% of the weight at day 7 post-infection. However, when mice is treated with the antagonist, when we block CCR3, then this reduction is only about 10%. So it means that blocking CCR3 can reduce the severity of the infection. And then again, we evaluated the presence of the neutrophils in the lung of those mice. And the first important data we have here is that at day 4 post-infection, we have the peak of the neutrophil infiltration to the lung. But then again, when we treat the mice with the antagonist, when we block CCR3, we have a significant reduction in the presence of the neutrophils in the lungs of those mice. And in line with these findings, we also observed that when we evaluate the expression of the receptor during the course of the infection, we found that the majority of the neutrophils that are present at day 4, they are expressing CCR3. So it's not just only the presence of the receptor, but sorry, the presence of the neutrophils, but also the presence of the receptor on the neutrophils. So it seems that the expression of this receptor on neutrophils is dynamic during the infection and is peaking when we have the big amount of inflammation in the lung. And we evaluated other inflammatory markers. In this case, I'm just showing you the expression of TNF-alpha, which is a pro-inflammatory cytokine. So you can see here how this is increased when we have infected mice with influenza. But again, if we block CCR3, we have a reduction on the expression of this cytokine. And then we wanted to know specifically if this might be like a global phenomenon in the lung, but also we wanted to know if this can also affect specifically the neutrophils. So we performed an intracellular staining for TNF-alpha in neutrophils. And again, you can see here in the histogram in red, the red line, we have these levels of production of TNF-alpha on the neutrophils. But when we treated the mice with the antagonist, then those neutrophils are producing less TNF-alpha. So overall, seems that when we block CCR3, we have a reduction on the recruitment of the neutrophils and therefore a reduction in the inflammation in the lung. Another effector function that plays an important role during acute influenza infection is the formation of those neutrophil extracellular traps. So here we evaluate the presence of the nets in the lungs of the infected mice by detecting mieloperoxidase and also citrulinated histone. So we have a background in the control mice, but then here you can see here this more bright signal for mieloperoxidase and for histone, and we have an increase in the area of nets in the lungs of influenza-infected mice. But when we block the CCR2 receptor, then again, we have a significant reduction on the nets formation. And also this has an impact on the pathogen load. In this case, we evaluate the presence of the influenza virus using the expression of the gene M. And as you can see here, we have this tendency in the reduction of the viral load. We don't think that it's like a direct effect of the antagonist on viral replication. It's more that because we have less inflammation, then the tissue has a better preservation, and then the viral replication is reduced when we block at CCR3. So far, I have shown that when we infect mice with influenza, then we have this massive recruitment of neutrophils to the lung. Those neutrophils upregulate the expression of CCR3, and this expression of CCR3 can increase some effector functions in the neutrophils, specifically the production of pro-inflammatory cytokines and also the production of NETs. But when we block CCR3, then we have a significant reduction of those neutrophils and therefore less inflammation and therefore a better preservation on the tissue. So that was what we found in the case of influenza infection. But as I mentioned earlier, it seems that CCR3 is not exclusive for either or bacterial or viral infections. It seems that, and all the reports that I have not the time to show, but have shown that whenever we have inflammation, then neutrophils can upregulate this receptor. And this is very important because while we need neutrophils to try to control the infection, then and overactivation of those cells then can be detrimental for the host. So in line with these findings, so we wonder whether this CCR3 could be important to protect the host, or what would be the role to protect the host, but now against the bacterial infections. And in this case, we move from Acinetobacter and then influenza virus, and now we tested infection with Streptococcus pneumoniae, which is a bacteria that is very important for public health, as it's one of the main causes of meningitis in kids younger than 5 years. So we established the model of infection in the lab with a mice, and again we infected the mice intranasally and then treated with the CCR3 antagonist, and we performed the analysis at day 2 and 4 post-infection. So one parameter that we evaluated in this case was the bacterial load. So at 2 days post-infection, the bacterial load is pretty similar in the two groups. However, at day 4 post-infection, you can see how the mice that are treated with the antagonist cleared more efficiently the bacteria. And again, we evaluated the neutrophils. We saw an increase of neutrophils at day 2, a little bit less in the case of the mice treated with the antagonist. But again, at day 4 post-infection, we have a significant reduction in the presence of neutrophils in the mice treated with antagonists. So it seems that it's a very broad event in which whenever we have inflammation, at least in several models in the lung, we are going to have this upregulation in the of CCR3 in the lung. And then again, here we analyze the tissue with hematoxylin eosin staining. Here you can see in the control mice the very well preserved structure of the lung. However, at day 2 post-infection, mostly with Streptococcus pneumoniae, you can see this increase in the cellularity in the tissue, but also this accumulation of cells that are like some focused pneumonia. However, in the case of mice treated with the antagonist, we have a significant reduction in the size of this puncti that we have on this accumulation we observe here in the mice that is infected and just treated with the vehicle. So far I have shown you here that CCR3 is expressed in the neutrophils during Acinetobacter, influenza, or Streptococcus pneumoniae infection. However, when we have done our analysis, our flow cytometry analysis, we observe actually that not only neutrophils express CCR3 in the lung when we are doing our different infection models, there are other cells that are expressing this receptor too. So among those cells that are expressing CCR3 too, we can find macrophages and monocytes. We can find these cells that are expressing CCR3 either in the valve, which means the alveoli, but also in the parenchyma in the lung. So yes, CCR3 can be expressed in other cells like macrophages and macrophages. So we are starting to explore these other populations, because also in the lung we have different subpopulations of macrophages. We have the alveolar macrophages, the interstitial macrophages, those monocytes that arrive to the lung because of the inflammation, and that they can differentiate either to macrophages or dendritic cells. So our preliminary results, again, showing that when we have an infection with Streptococcus pneumoniae, we have an increase of the presence of the macrophages. This is just general macrophages. We have not done the very specific analysis on the different subpopulations. And the presence of those macrophages are reduced at day 2 post-infection. So again, it seems that CCR3 not only has an impact on the neutrophil presence and neutrophil activation, but also in the macrophages. We also find presence of the CCR3 in the macrophages during influenza infection. So we are trying to also characterize those subpopulations and trying to evaluate what is the impact of the expression of CCR3 in those different macrophage subpopulations. So what would be the take-home message from the talk today is that we have revisited CCR3 because we have gone from the eosinophil associated receptor in the context of allergens mostly to the point that we think that CCR3 is a broader regulator of pulmonary myeloinflammation. And this is important because it has consequences in both in the pathogen control, and it doesn't matter if it is a virus or a bacteria, and also on the tissue injury, which it's going to reflect on the host. So basically, we show that CCR3 is dynamically expressed during respiratory infections. It seems that regulates the myeloid cells recruitment and function, at least for neutrophils. We are going to evaluate for macrophages. And this might represent a broader regulatory node linking the chemokine, the recruitment, the activation of the cells, the pathogen control, and the immunopathology of respiratory infections. So we actually are wondering like how CCR3 determines the quality and the outcome of the inflammatory response, because inflammatory response is made to control the infection, but if this inflammatory response is exacerbated or is not of a good quality, then it's gonna be detrimental for the host. So with that, I would like to acknowledge the people, all the people in the lab. This work has been done mostly by Fatima, Jesus, and David. Also our collaborators at UNAM, but also at other institutions in Mexico City, the University of California, and the funding. And with that, I will be happy to take questions at the end. Thank you very much.
Dr. Aracely, thank you very much for this excellent presentation and interesting science. Now we will not continue with the second speaker, but before that, I need to remind you, the audience, to please submit the questions through the YouTube chat, please. And now we can continue with the next speaker, which is Dr. Otoniel Rodríguez Jorge. Dr. Otoniel Rodríguez Jorge is a professor researcher at the Centro de Investigación en Dinámica Celular of the Universidad Autónoma del Estado de Morelos here in Mexico, where he leads the Cellular Assisted Immunology Laboratory. He earned his PhD in science in cellular and molecular biology at the same university, with focus on system immunology under the joint supervision of Dr. Maria Angelica Santana and Dr. Denis Sefi of the École Normale Supérieure in Paris. I think that's French. His research integrates experimental immunology with bioinformatics, computational biology, system biology, and functional genomics to understand the molecular mechanisms that shape human immune response. A major focus of his work is neonatal immunity, T-cell activation, immunometabolism, and the influence of maternal and perinatal factors of early immune reprogramming. He also serves as coordinator of a bachelor program in science in biochemistry and molecular biology at the UAEM. Today, he will present the talk entitled Programming T-cell Responses at Birth, Perinatal Regulation of Neonatal Immunity. Dr. Rodríguez Jorge, thank you very much for being with us, and please go ahead.
Hello, everyone. I hope you can see me, hear me.
Perfectly.
Okay, perfect. So first, I want to thank the organizers, and I want to thank ALACI Junior community for the invitation. And so this is the topic I want to talk this afternoon. So first, I want to give you a very short introduction of the topic. So this topic is a little bit extensive, but I want to first talk to you about the neonatal immune system. So there's a lot about innate cells in neonates that have been published. We know now that many neonatal cells from the innate system have reduced functions, but this time I will be focusing on T cells, which is the main topic of our lab. So in the lab, in the last years, we have been describing CD4 T cells, CD8 T cells, and by this time we know now that CD4 T cells and CD8 T cells have reduced activation and also have a bias differentiation towards a Th2 and Treg response. Also, CD8 T cells have shown an innate response and a reduced cytotoxic activity, and we have been able to track these responses to the epigenome, which is responsible for a characteristic transcriptome for these CD8 T cells. And we know that T cells play a key role for defending the neonates against all the possible infections that may be causal of infections in the neonatal period. So neonates are very susceptible to many infections, and also we know that CD4 T cells may be may have a different program. So in the last years, we also characterized the transcriptome for these cells, and this is work done mainly by our postdoc, which is Linda Kempis. She was able to track this transcriptome. We found that neonatal CD4 T cells have a characteristic transcriptome, which is mainly enriched in pathways like metabolic pathways, also intracellular signaling pathways, and many others. But this also is represented or overrepresented in pathways like cell cycle and not in the characteristic typical, let's say, pathways found in adult cells. So we found that actually these genes can form a typical or a characteristic protein-protein interaction network, which I can show you here, that seem to be controlling all the program in neonatal CD4 T cells. And we believe that this pathway, this network is very relevant for controlling the neonatal CD4 T cells. And this is also present in basal conditions, but we later found that upon activation, these cells also overexpress these same genes among others. So here I show you that upon activation with TCR and CD28 signals, neonatal CD4 T cells actually respond as we can expect, but they don't do it in a typical way. They respond in a different way where they overexpress the same genes that we found in the basal conditions, let's say the HIF pathway, the glycolysis pathway, RAS pathways, and many others, but also they overexpress other metabolic pathways. And this is in contrast with the adult CD4 T cells, where they overexpress the typical characteristic pathways like cytokine interaction genes, and also toll-like receptor and many other signaling pathways. So in a way, our CD4 T cells from neonates are responsive. They can do many things, but they are not our typical CD4 T cells, let's say. So we went further and we tried to characterize what were doing these cells. So we explored the cytokines that they were producing, and we found that they were able to produce in very small amounts these cytokines, the typical TH cytokines, but not in the typical amounts found in adult CD4 T cells. So for example, interferon gamma, we expected already that it was not going to be produced because of the antecedents. But the others also, they were not produced in high amounts. We also found that they were actually proliferating much more than adult cells. This was novel for us, and it was also predicted by the transcriptome. So we were able to kind of find a whole characterization of neonatal CD4 T cells like we did before for CD8 T cells in neonates. And this was done before I was a researcher in the lab. I was already a student, but now we found that neonatal CD4 T cells were responsive. They were doing a lot of glycolysis. It seemed uh, because of the transcriptome, they were proliferating. They activated, but not in a typical way. It seemed like more tolerogenic profile because of the genes they were expressing, like PD-1 and many other genes. And this was a profile that was present in basal conditions, but it was enhanced upon activation. And also later, we wanted to corroborate this profile and go further. So another student in my lab characterized the metabolic functions of these cells. So he found that neonatal CD4 T cells overexpress GLUT1 in the membrane. Also, they were consuming more glucose before and after activation, but they also uptake more fatty acids. And this way, we kind of understood that neonatal CD4 T cells were actually more active metabolically. And here I don't show you, but we were also able to track this or to correlate this with more lactate production. So this was not only nutrient uptake, this was actually metabolic activity. So what we found in the transcriptome, it was actually translated into metabolic activity. And also there was a paper which corroborated this with an indirect this technique that is translation inhibition. So Francisco also compared all the metabolic enzymes in activated CD4 cells between neonates and adults, and he found that actually it was not only one enzyme or not only the transported of glucose, GLUT1, it was actually most of the glycolytic enzymes. So really the glycolytic activity was modulated by most of the enzymes of glycolysis. So in summary, for these cells, we know now that upon activation, we have more GLUT1 translocation in the membrane. This is correlated with more glucose consumption, lactate production. Also, we have more fatty acid uptake, and we are exploring other metabolic markers, but these cells are more metabolic active, and they are very responsive, just not in the typical way that we expect for a CD4 T cell, which is cytokine production and many other things. So after this, we started to think about the things that may modify or may contribute to the programming of the neonatal CD4 T cells. And we know it has been reported that there are many perinatal factors that can actually contribute to the programming of neonatal immune system. So there are many fetal factors like placental abnormality and many other things. There are maternal factors that can contribute to the immune system of the neonate, environmental factors, epigenetic, genetic, immune and inflammation mediators. There are also maternal stress, fetal stress, and many other things that may contribute or can contribute. Some of them have been proved to modulate the neonatal immune system. Some others have been hypothesized. So what we actually know is that preterm birth has been associated with maternal and perinatal factors, like not only infections, but also inflammation without infection, maternal and fetal and neonatal inflammatory response have also been identified in preterm birth, where interleukin-6 has been reported as a key factor, meaning that if interleukin-6 is high, usually it has to be with a preterm birth. It's highly associated. Also, cesarean section has been associated with an increased risk of allergic diseases later in life. And this is very, very important because we have data from recent years that in Mexico and many other Latin American countries, that cesarean section is the main mode of delivery in these countries. In Mexico, actually, these later years have been like 55% of the births. And in the case of preterm births, most of them are cesarean sections. So this is a very important topic to address. Regarding the actually immune system in these two factors, it has been reported that CBMCs from neonates born by cesarean section show reduced immune responsiveness. This has been published. So there are not many studies about direct measurements of immune activity in just at birth because of the difficulties of the system, because it's not the same to do the study in for example, in mice than in human, and many other things because of the ethical and many other aspects. So because of this, we wanted to perform this study, and in the next slides, I will be talking about our results that we published in this paper. So again, Linda performed a transcriptomic analysis where we compared 3 groups of neonatal CD4 T cells, cells that came from births from vaginal delivery, preterm births, where they were cesarean sections, and cesarean sections, but from full-term births. if you see here our PCI analysis, you can see that the main difference will be between vaginal delivery and cesarean section, not with regard of preterm. So here in the Vulcan plot, I show you our differentially expressed genes, and here I show you our pathway analysis with these differentially expressed genes, and most of the pathways are annotated in the vaginal delivery CD4 T cells. So these are pathways related with immune responses like TNF signaling pathways, NF-kappa B signaling pathways, and many others. Of course, we went to see each of the genes, and I can tell you that they are actually associated with an immune response, like interleukin-1, NF-kappa B, YUN, and many others. So neonatal CD4 T cells from vaginal delivery neonates have a characteristic transcriptome, and this is a signature for this group of neonates. So we try to characterize more this group of CD4 T cells, and our gene set enrichment analysis told us more about this. And we found that cesarean section cells are more, have genes more related with RNA processing, while vaginal delivery cells are more related with immune processes. Preterm cells are maybe proliferating or something like that because they are they have processes like say cell cycle progression. Again, we did our protein-protein interaction network, and we were able to find two main networks, one related with cell cycle and one related with immune related processes. So we have these two processes. We went further and we use as an analysis to find all the transcription factors that were activated, that were active during each of these processes. And we found that during vaginal delivery, there were this group of factors that were active, and they were related with an immune response. You can see here YUN, MYC, NF-kappa B, STAT1, IRF1, you can see many of them. Of course, the other groups have also some transcription factors. None of them are related with an immune response. When we went to see the score of these transcription factors, only the ones related with the immune response and the vaginal delivery group were significant. So this is a result that we trust. Then Carlos went to validate this analysis in a different group of cells. So he purified again CD4 T cells, and he measured activation of NF-kappa B and AP1 by evaluating the phosphorylation of P65 and c-Jun upon activation. And in these conditions, we found more activation of these two transcription factors in the vaginal delivery group and also in the cesarean section group, but not in the preterm group. Also, trying to validate some of these results, Carlos evaluated the proliferation of the cells in these three groups, and we found that vaginal delivery cells didn't proliferate in great proportion, but cesarean section and preterm cells did proliferate a lot. So these two are more proliferative than vaginal delivery cells upon stimulation. After that, to complete our characterization of the responsiveness of these cells, we evaluated the production of all the Th cytokines and also other molecules that we consider relevant for neonatal responses. And these were granzyme B, perforin, and innate cytokines. And here I show you the heat map. So as you can see, many of them were produced in the preterm group, also somewhat produced in the vaginal delivery group, but not much produced in the cesarean section group. But I will show you the mean production, the average production. So here in the panel A, I show you the average production of each group, and also I show you the production in the adult CD4 T cells for comparison. And you can see that preterm cells, they produce quite a lot of all the cytokines. Here only, I hide only the cytokines that were not produced from all of our panels. And this is produced in the medium. We use the panel, the BioLegend panel. So this is actually produced in the medium. And preterm cells actually produce more than even than adults. Cesarean section, they don't produce a lot of cytokines, and vaginal delivery cells, they produce somewhat a certain amount of cytokines. For comparisons also, we obtain an average of cytokines by profile. The Th1 profile, the cytotoxic profile, the innate profile, etc. And we did this radar graph. So I show you that every, so I show you the average production for each group, and you can see that the preterm cells, they produce more of each profile. After that, we find the adult cells in red, and then you can find in blue the vaginal delivery cells, and after you can find the cesarean section, which they don't produce a lot of cytokines. So T cells from vaginal delivery neonates are more responsive than those from cesarean section, while T cells from preterm are more inflammatory than those from full-term neonates. And actually, these numbers of cytokine production is actually very significant because it's more than the production of adult CD4 T cells. So we have now a working model for our CD4 T cells. We believe that during birth, we have this kind of hypothesis of what is happening. We believe that for CD4 T cells, this is important to clarify. For CD4 T cells, before term, preterm CD4 T cells are already mature enough to present a highly inflammatory profile. They are very responsive, and once they approach the term by 36 and 37 weeks of gestation, they approach a less responsive profile. And if you are born by cesarean section, then you will not have responsive CD4 T cells. But if you are born by vaginal delivery, then all the process of vaginal delivery may subject the CD4 T cells to signals that can program these cells and then that can activate these cells maybe by a different way, so that these cells can have a more responsive profile. So we call this characteristic of vaginal delivery CD4 T cells a vaginal delivery associated activation. And this is responsive for the difference between CD4 T cells from vaginal delivery and cesarean section neonates. So for this work, I want to thank all the members of my lab. I also want to thank Linda Kempis and Angelica Santana, because they are the two people that actually began to study all the transcriptomes. Also, Salvatore Spicuglia, he is our collaborator in France. And there are many people that participated in this study. Carlos, here he is. He's the PhD student who is going to graduate soon, and he's responsible for this paper. But these people also participated a lot. And I want to thank also the mothers and the babies that donated the samples, the institutions that gave permissions to do this study that is very difficult to do without permissions, and they are very difficult to obtain. And in the case of preterm babies, it's a hell to to obtain these samples, but they are very important, because otherwise we will not be able to do this kind of studies. And I want to clarify that these results are for CD4 T cells. We are also doing this for CD8 T cells. And as you know, CD4 T cells and CD8 T cells are very different. And what we have found in neonates is just that CD4 T cells and CD8 T cells are very different too. And this time I will not show you anything about CD8 T cells, but I will tell you just that the story is also very interesting and very different. So I just want to keep you interested for our next story that I hope to publish soon. So thank you, everyone, and I will be here just to respond your questions.
Thank you very much for this very interesting science, Dr. Otoniel. I would now like to invite both the speakers to join us for the discussion. We have collected some questions from the audience through the YouTube chat. I will begin with the first one. I think these are questions to Dr. Aracely. Let me see. Dr. Isabella Galbao asked to Dr. Aracely, has the treatment reduced the number of neutrophils involved? Have you checked if the treatment improved lung function?
We have not done those type of experiments like expired volume or respiration resistance, like those physiologic studies, no. But we expect that as those like mice have reduced inflammation, they could eventually have a better function after recovery of the infection. Actually, we have some preliminary results in which we just kept the mice to recover and clear completely the virus. And we observed that the mice that were treated with the antagonist, they gained more weight more rapidly in comparison with the mice that are just mock treated. So we expect that lung function would be also better on those mice that were treated with SV, with antagonist.
Amazing. Thank you for that question. We also have a comment and a question for Val Dutra, the ALACI president. She says, great talk, Aracely, because CCR3 has more than one natural ligand, CCL11, 24. I was wondering if the neutrophil activation via CCR3 is mediated by one of these particular ligands. That's the first one. Also, it is activity of CCR3 in different cell population, neutrophils, macrophages, mediated by the same ligand or by a different one,
Yeah. For neutrophils, we just have tests so far CCL28 and CCL11. We have not checked the CCL24 or CCL26. And what I didn't show the data about the chemiotaxis assays. It seems to have like a very few chemotactic activity, at least in our transwell analysis, CCL28 and CCL11. But if you remember the data, it seems that CCL20 has, it's more powerful to say like that, to improve bactericidal activity in neutrophils than CCL11. So both improve bactericidal activity, but CCL28 has better bactericidal, like improve it better than CCL11. And for macrophages, we have not done any functional experiments like activating or stimulating the cells with the ligands, because we just starting with like characterizing the expression of CCR3 on those macrophages during infection. And another thing that I think is going to be important regarding to the ligands is either depending on the infection, one or another ligand is produced, and in the context of what ligand is
I think it's her.
Okay, I think we're having a problem with Aracely's internet. So if you, in the meantime, we can continue with the next question. This question is for Dr. Otoniel from Bio UNAM 2011 that I really, I know who this person is from Eugenio Contreras.
Okay.
He's asking you a question about your research. He asked, what about work? And have you seen any difference between the sex of the baby? Any changes, long-term CD4 T cells from the mother after birth?
Okay. We have actually looked for differences between female and male in CD4 T cells, also in CD8 T cells, and we did not find any differences based in sex. So no, but maybe in other cell types, it's possible that there are differences, and not in this case. And regarding long-term effects, it is possible. We are looking for this, but it is a very difficult project to do. But we are actually trying to do this work.
Amazing. Thank you for that answer. Hopefully that's enough. We have another question for Val. Val Dutra says, thanks, Otoniel, fantastic talk. What do you think is the mechanism by which cesarean reduces immune response or vaginal delivery increases it?
Well, my hypothesis is that vaginal delivery enhances the responsiveness. This is what I believe is happening. And there are many possible ways to do this. I believe that a possibility is that signals during labor can contribute to enhance T cell responsiveness, but not only CD4 T cell responsiveness, also CD8 T cells and many other immune cells, because during labor, there are signals, stress signals, cytokine signals, many signals. And it is very possible that all these chemicals can modify and can program, at least temporarily, the responsiveness of the cells. Not necessarily in a bad way. Maybe it is what it is. It has to be that way, biologically programming the baby for the next period of the life. And it has been reported, for example, I don't remember the name, but it's the activation of the cell when it is not it's not involved the TCR. I don't remember right now the name, but it's bystander activation of T cells. I believe it is that activation. So it's a small activation by many signals not involving the TCR.
Okay, we have an additional question for you, Otoniel, but also ask if you think bacteria may play a role.
In our case, I think it is not possible. I think we discarded this effect because we use cord blood, and it is before the placenta it is expelled. So I think this is not involved. And with regard the preterm births, we also use samples from mothers that are not sick, that are not infected. It is very difficult because many preterm births is related or associated with infections. And sometimes many of them are not detected. So it's difficult to do and difficult to assure, but we kind of prove in the paper that this is not the case. But of course, if microbiota is involved, it will have an effect for sure. But these effects that we are reporting in this paper are not related with microbiota or other bacterial infection.
Amazing, thank you for that
Yes.
answer. I think that we don't have more questions, so I would like to say thank you for the presentation, wonderful science. But before close the session, I would like to make some announcements. Let me share my screen. It's really quick. Okay, can you see my screen now? Let's say yes.
Yes.
So the first announcement that we have today is, please, we are very pleased to invite the community of the ALACI and the SBI meeting, which will be take place in Ouro Preto, Brazil, from October 18 to the 22nd next year. Please stay tuned for the preliminary program and additional information. Probably we will have some additional information during the next meeting, the next seminar. And about the seminar, for the next month, we have two wonderful invited speakers, Dr. Eloisa Arana and also Dr. Luisa Duarte, who's gonna discuss fantastic immunology science next month. It will be September 17. So hopefully, guys, you can join us. You can discuss the science that we are excited about here in ALACI. And essentially, those are the important announcements that we have before the closing. So thank you very much. Oh, before that, as you can see here in the screen, also I would like to thank to the organizing committee, that is Isabella Gabao, who is our ALACI Junior Community IOIS representative, Sandra Ortega. She is also ALACI Junior Committee, and also Maria Julia Lamberti. She is not here today. She couldn't make it today, but she is usually helping us to organize these seminars too. So that's essentially the end. Thank you very much, Dr. Perez. Thank you very much, Dr. Rodríguez, for sharing your work with us today, for the stimulating discussion. It has been a pleasure have both of you part of the ALACI Seminar Series. I will also thank to everyone who joined us today from across to Latin America, the Caribbean and beyond, and also makes the people that made some questions. Before we close, please remember complete the attendance and feedback form shared in the YouTube chat. That will be helpful for us. The feedback is valuable and help us to improve future editions of the seminar. The recording of today's seminar will remain through the ALACI Junior Community channels, and essentially follow us in the next ALACI Seminar Series. Thank you very much.
Thank you.